Hello, I'm Larry Reiter, and I'm a professor in the Department of Neurology at the University of Tennessee Health Science Center. I really appreciate this opportunity to talk about our dental pulp stem cells, and I hope today to tell you about a couple of our particular stories, but also to give you an idea of how we can use these dental pulp stem cells to study rare neurogenetic disorders. So what are dental pulp stem cells? I myself was unfamiliar with them until I started working with them. And it turns out there are these neural crest-derived stem cells that are inside the tooth cavity, and they have multipotent potential, including the ability to make neurons and glia in culture. What's really nice about these cells and we take advantage of this, is that they're inside of shed baby teeth. So children lose their teeth naturally, as you can see in this photo here. And we can actually remotely collect those teeth to make dental pulp stem cells. Here is a kit that we use that we send to families, and it includes a consent form, a tube of media, a questionnaire called the SEQ that allows us to remotely assess autism features, and a return box. And so if we get contacted by a family, we can very easily collect teeth remotely for these rare disorders. So the process is the parent will email us. We need to see a genetics report. And then once we have that report and we know that they have a particular syndrome, that's one of the ones that we're collecting, we'll send a tooth kit out. And whenever the tooth falls out, or if the tooth is extracted, they'll send the tooth back to us. In the lab, we'll crack open the tooth and we'll mince the pulp and grow out the dental pulp stem cells. So this has been very successful for us. We've collected over 200 dental pulp stem cells from 200 individuals. And we've had a big emphasis on the 15q region, which I'm going to talk about today. So we have a lot of individual cell lines from Duke 15q, Prader-Willi, and Angelman syndromes. But we also collect other disorders, and we've been in the process of building this collection in particular directions depending on where the research focus is. So the process after we make the dental pulp stem cells to differentiate into neurons is very simple, actually, and that's the point, is we don't reprogram these cells. We just coax them into being neurons. That does require that we reset the epigenetic landscape a little bit. And then we add factors to the media that promote neural differentiation. So these stem cells are then promoted into a neural lineage and then matured as neurons for 4 or 5 weeks, depending on the experiment. And then we can do molecular studies and cellular studies that I'm going to show you in a minute. So what do they look like? So on the left, you'll see a bright field image of dental pulp stem cells. On the right, you'll see a bright field of DPSC neurons. You can see these long processes in culture, and they look pretty good. They look pretty healthy. When we stain them, you can get a better idea. So the actual dental pulp stem cells look like sort of a fibroblast-like morphology, whereas the differentiated neurons have this neuronal look, and they have a nice shape to their soma. This is maybe not the greatest photo, but we have some others that I'll show you. And basically, we're able to culture them for several weeks, but they're very immature neurons. So you might be asking yourself, what are these things and what markers do they have? This lab in 2009 actually was one of the first labs to make neurons from dental pulp stem cells, and they could get currents from these neurons. They could also look at a variety of markers, including stem cell markers like vimentin and nestin, but also neuronal markers, neural tubulin, and glial markers like GFAP. So we've actually been able to repeat some of these markers and also expand the cohort of markers into GABA-A, receptor and MAP2 and neuroligin 1, which is in DPSCs and neurons. However, as I'll show you, not only is the morphology right, but we do see a lot of markers you would expect to see. And we've done some studies that were on specifically looking at these markers in the differentiated DPSC neurons. So one more thing I wanted to point out is we did a study with Janine LaSalle's lab, and we looked at the whole methylome across a variety of stem cell types or cells that people typically work on in the lab as representative of the particular disorder. So iPSCs you might be most familiar with, but people use lymphoblasts. They like to use this SY5Y neuronal line. There are several other embryonic stem cells here, and then some methylation studies of cortex and placenta. And what you'll notice if you look carefully is that the dental pulp stem cells, they actually have a global methylome pattern that matches the placenta quite well, a lot better than the iPSCs or even the embryonic stem cells. So it's got this sort of similar pattern. And in fact, when you look whole methylome, the placental cells do overlap with the dental pulp stem cells, whereas the SY5Ys and the iPSCs, not so much. So it's not a perfect analysis, but it does indicate that we're looking at something that may be more representative of the actual state of neurons in a particular syndrome. And in this case, we looked across all of these individual DPSC lines. So just to give you some additional information, yes, we can make other cell types out of DPSCs. We've done it in our lab as well. We can make chondrocytes and adipocytes, and other labs have done this. And in fact, for a very long time, people were using them to attempt to regrow tooth material, which is not really what we do. We make neurons, but we're able to do this as well. All right, so that said, let's dig into what we've done with DPSCs in our lab and give you a sort of a flavor for how to use dental pulp stem cells to study different neurogenetic syndromes. Specifically, in this case, I'm going to talk about 15q syndromes. So this is just a brief overview for those of you unfamiliar with these particular syndromes. The first one I'm going to talk about is called Prader-Willi syndrome, and it's characterized by hypotonia, failure to thrive, developmental delay, hyperphagia, cognitive defects, a little bit of dysmorphology. But what I'm going to emphasize is some work that we've done where there happens to be an increased risk of autism spectrum disorder in the uniparental disomy cases. And these cases are maternal uniparental disomy. We'll get into those details in a bit. So the majority of cases are caused by the loss of paternal 15q11.2 to q13, and there's several genes that may be involved, and I'll get into the details again as we progress. The frequency is approximately 1 in 15,000 births. So a related disorder at the genomic level, but not at the phenotypic level, is a syndrome called Angelman syndrome. Also characterized by hypotonia and developmental delay, but there are very specific phenotypes involved in Angelman. One of them is an ataxia and a lack of speech and communication difficulties. There's also, as you can see, a very happy demeanor among kids who have Angelman syndrome, and they can easily be excited. So this is actually caused by the same deletion, but it's an imprinting disorder. So it's caused by the maternal inheritance of that deletion, whereas Prader-Willi is caused by the paternal inheritance. And again, I will get into the details of that in a bit. Again, you might not be surprised that the frequency is approximately 1 in 15,000 births. So a syndrome that I've worked on quite a bit, even clinically, is the duplication 15q syndrome. And it is also characterized by failure to thrive and developmental delay, hyperphagia, cognitive defects, a little bit of dysmorphology, and an increased risk of autism spectrum disorder. Especially that was our focus when we studied these kids, which are all interstitial duplication cases. So they actually have one extra copy of the region. And as you get more copies of the region, there tends to be an increase in the epilepsy frequency, and it's a very severe epilepsy in kids that have isodicentric duplications of 15q. Again, now we're talking about the same region that was deleted in the previous two syndromes, but is duplicated in isodicentric 15q. And again, the incidence is about 1 in 15,000 births. So what does all this mean? So the long and short of this story is that you can get Prader-Willi syndrome several ways. One is to get a deletion of 15q on the paternal allele. One is to have maternal uniparental disomy, so two maternal copies, two active maternal copies. And the other is to have an imprinting defect that causes the expression or lack of expression, in this case, of the paternal genes on the paternal allele. In a similar manner, Angelman syndrome can be caused by deletion of the maternal copy, paternal uniparental disomy, so two copies of the paternal allele, or an imprinting center defect, or finally, a maternal mutation in the UBE3A gene. The duplication cases are caused by a duplication. The interstitials are the ones that I showed you, and they have 3 copies of the region. The isodicentric duplication cases, there are 4 copies of this region, 3 of them being active on the maternal chromosome if we're talking about UBE3A. And then there's paternal interstitial duplication cases that have a slightly different phenotype, and we're not going to really talk about those guys today. All right, so let's talk about the region. The region that we keep talking about is between breakpoint 2 and breakpoint 3 here. This is the critical region for Angelman syndrome and for Prader-Willi syndrome and for duplication 15q syndrome. This is the region that is deleted in the majority of cases of either AS or PWS. And the genes that we're talking about in the case of Prader-Willi are these paternally expressed genes here. So these are genes that are only expressed from the paternal chromosome in neurons. They include MAGEL2, SNRF, SNRBN, and you may have heard that there are some cases where maybe it's possible that the SNO RNA is the primary cause of this syndrome, and that's also an imprinted set of genes. In the case of Angelman syndrome, we're talking about maternally expressed genes, which are actually, there's only one, and that's UBE3A. So UBE3A is expressed in neurons from the maternal chromosome and not the paternal chromosome. We won't get into the details of imprinting, but you need to know sort of the landscape here to know what we're going to talk about in terms of autism and other phenotypes that we see. This is just to show you that we did, in this 2018 paper, we did some FISH validation, which was kind of nice that we used our dental pulp stem cells for. So in the case of Angelman syndrome, in this cell line, you can see there are 2 red dots and 1 green dot, the green dot being the SNRBN gene. So they're missing a copy of SNRBN on one of their chromosomes. And then in this cell line, you can see that there are 2 red dots and 2 green dots. This is a control line. And in this IDIC case, you can see that there are 1, 2, 3, 4 red dots and 1, 2, 3, 4 green dots. So this is a case of having an isodicentric duplication, an external chromosomal piece that has 2 copies of this region on it. So in that study, what we did was we did gene expression. And the first thing we looked for in our RNA-seq data using control, dup15q, and Angelman deletion lines was what is happening in the 15q region. When we look in the 15q region, we actually found a significant trend that related copy number to gene expression for 2 genes, PERK2 and UBE3A. And we expected UBE3A. HER2 actually is a little bonus there because actually HER2 is a ubiquitin ligase as well. It is in the critical region and it is able to activate UBE3A. So there are some thoughts here that maybe HER2 is involved in some of the phenotypes as well. But the real meat and potatoes of this study was to look at gene expression outside of the 15q region. And when we did that, you can see here in the DPSCs, we didn't really see much signal, but on this heat map, you can see that there was a decrease in gene expression in the duplication cases and relative to deletion and both deletion and control. So that was a critical result that showed us that there was a signature for DUP15Q that was really unique in DPSC-derived neurons. In the Angelman cases, not so much. We really didn't see much of a signal in terms of unique gene expression. But when we looked at the duplication versus deletion, we did see in neurons a set of transcripts that are down in duplication and up in deletion, or up in duplication and down in deletion. These were very interesting transcripts. So we next looked at our DUP15Q neurons and compared gene expression to ASD brain and 15Q duplication brain RNA-seq studies that other people had done. And we found some very interesting overlaps. So one of the things we found in the end was a gene called FOXO1. Now, FOXO1 is of interest to us because FOXO1 regulates another gene called RORA. RORA is an ASD-associated gene, and it is significantly downregulated at the protein level in IDIC15 neurons. So that was the real conclusion of our paper, was that when we then took a separate cohort of IDIC15 kids, we could see that there was a downregulation of RORA in IDIC15. Okay, so now we have sort of a picture of what we were thinking in terms of ASD and UBE3A expression. So in the case of Angelman syndrome, this is loss of maternal expression of UBE3A. This is not associated with autism. It's more associated with cognitive defects, but also communication defects. And then paternal UPD, you would expect to have not really a huge increase in UBE3A. As we get further up above the normal range, you would expect to see expression in maternal UPD cases that is higher than the normal range of UBE3A expression. And in the case of interstitial duplication, you would expect to see more expression even than the UPD cases. And in the isodicentric duplication cases, again, you would see even more increased expression of UBE3A. So this is our overall view of the association of UBE3A and ASD. And so what I'm going to talk to you about next are these Prader-Willi maternal UPD cases and some interesting things we found that are related directly to ASD. So to remind you, now we're still talking about the same region, but now instead of talking about UBE3A so much, we're going to really focus on these genes and the paternal specific expression or loss of paternal specific expression in the case of Prader-Willi syndrome. Okay, so one of the things that got us interested here in looking at ASD and Prader-Willi was that about 35% of the UPD patients meet the criteria for ASD. So this is a subset of Prader-Willi, right? The majority of those cases have deletion, but there is a subset of Prader-Willi UPD kids that have an increased risk of ASD over the deletion patients, right? That was of interest to us. We wanted to find out why they have an increased risk for ASD. So as I said in the beginning of my talk, we use the social communication questionnaire, and this is a validated parent questionnaire that we use to assess ASD remotely. Scores above 15 indicate possible ASD. What's interesting is as we looked across all of our samples, so these are all of our Prader-Willi DPSC lines and all of the individuals involved, we see that about 33% of the Prader-Willi UPD cases have an above 15 score indicating possible ASD. So moving further on, we designed an experiment to do gene expression analysis and focusing on the Prader-Willi UPD plus versus minus ASD, but also on Prader-Willi deletion versus control, and all of these classes of Prader-Willi syndrome versus control. So we set it up by using 4 individuals per group. We differentiated the DPSCs into neurons, and then we did RNA-seq on each sample. So first things first, we looked across the duplication deletion region, the Prader-Willi Angelman critical region that we've discussed about, and we wanted to see if there were any indicators of gene expression, for one thing, that said that we were looking in the right direction. And the first thing we notice, of course, is that HERC2 and UBE3A are up in UPD cases, down in deletion cases, and in about the middle in the control cases. So not a really big surprise there. But what's really an indicator that we're on the right track here is that these blue genes, which are all paternally expressed genes that are associated with Prader-Willi syndrome, these are down in both deletion and UPD cases versus control. So that seems to indicate that our imprint is there, and the cells that we are analyzing are representative of the syndrome. Okay, so what do we find? So when we looked across these different classes versus control, so we looked at deletion, UPD plus, and UPD minus versus control, we found 12 transcripts that are shared among all of these classes of Prader-Willi syndrome. And they are interesting genes. So some of them turn out to be involved in neurodevelopment. They're involved in neuropathy. Some are involved in autism. These are all interesting genes on their own. We have now done gene expression, obviously, by RNA-seq, but we've attempted to do protein analysis as well. And in some cases, we do see changes, but at the moment, we decided that this wasn't really our primary focus in the study. And so we sort of left it at the gene expression level, but you can see that some of these genes are clearly different in all of the Prader-Willi groups versus control, and that they're of interest in general because of their, in particular, this gene SNTB2 is involved in the regulation of secretory granules. So again, it may be associated somehow with MAGEL2, which is one of the key paternally expressed genes that is lost in the deletion cases. All right, but what really struck us and what we were really looking for was the Prader-Willi UPD plus ASD versus the other groups, specifically versus the minus ASD group. So if we looked at Prader-Willi plus ASD versus minus ASD, we found 312 transcripts that are dysregulated somehow in the plus versus minus ASD groups. What was really striking to us, though, was that when we did David enrichment, which you may or may not be familiar with, we saw a huge enrichment score for mitochondria, mitochondrial inner membrane, mitochondrial electron transport chain. So all of these are mitochondrial hits. So we went to dig deeper and to figure out, is there an association now between plus ASD and a mitochondrial defect? So if you look on a heat map at these transcripts, you find that, again, there is a dramatic difference. There is a downregulation, a huge downregulation of all of these transcripts versus the minus ASD group, and as well versus the deletion group, and somewhat versus the control. But specifically, we're looking at plus ASD versus minus ASD here, and we see a very dramatic difference. So we then decided, all right, if we see a mitochondrial defect, how can we analyze that particular defect? And we just went looking. We used a marker called TOM20. That's an outer mitochondrial membrane marker. And lo and behold, when we use beta tubulin to outline the neuron, and then we use TOM20 to look at mitochondria, we see here in the minus ASD group that this TOM20 staining is nice, evenly spaced in the soma and out in the processes. However, in the plus ASD group, we see these inclusions and these aggregates that are sort of perinuclear aggregates of TOM20 staining. So we wanted to try and dig a little deeper into this phenotype. This was a nice, striking cellular phenotype. So what we did was we used confocal imaging combined with Ameris to outline the area of the neuron that we want to look at using beta tubulin, and then to analyze the coverage of the TOM20 staining using the green channel, and compare the two groups, the aggregated mitochondria versus dispersed mitochondria. This was done in a blinded fashion. So we collected, I had a technician who would fix the neurons, and then my graduate student would do all of this analysis without knowing the genotypes of the cell lines. So in the end, we did this unbiased analysis to look at mitochondrial coverage, and we found, using 6 individual cell lines per group and 15 neurons per individual, that there was a significant difference in the Prader-Willi UPD plus ASD area. It was decreased versus the minus ASD, the deletion, and the control. So this was a striking, quantifiable result from a striking cellular phenotype that we identified. And now this paper is out in Frontiers of Molecular Neuroscience, and you're welcome to take a look at it. But in the end, we felt like this was a firm connection now between Prader-Willi UPD plus ASD and mitochondrial defects. So why are we interested in that? So as it turns out, mitochondrial defects have been associated with ASD before, and there have been publications showing reduced mitochondrial expression across brain regions in postmortem ASD brain, as well as mitochondrial dysfunction that's associated with ASD just in general ASD in the population. As well, these mitochondria help maintain ionic gradients and calcium homeostasis at the synapse. So there's functional consequences to defects in mitochondria that are beyond just energy metabolism. And disruptions in neuronal mitochondria function or number can disrupt these ionic gradients and lead to defects in neurotransmitter release. So our paper really tied these together and showed a significant decrease in mitochondrial volume and the appearance of these mitochondrial aggregates, which we're still unsure about, in neurons from PWS, UPD plus ASD individuals. So now the next step is, what do these aggregates mean? Are these mitochondrial dead or dying? And are there functional consequences to that in terms of energy metabolism in these neurons? Okay, so I showed you what we could do with gene expression. Now I'm going to show you 2 other things that we've done with our DPSC neurons. And the first, actually both of them are still in process, but the first one we've been working on for quite a while now, and that is sort of individual cellular circadian rhythm. These are actual traces from control DPSC neurons that show a luciferase signal for either BMAL or PER2. And if you're familiar with the circadian cycle, you know that BMAL and PER2 show this opposite pattern. One is up and the other is down, the other is down, the other is up. So you expect to see this nice rhythmic cycling, and that's what we do see. However, when we look at Prader-Willi syndrome and a related disorder called Chaff-Yang syndrome, where they have truncating mutations in MAGEL2, you can see that the Prader-Willi syndrome neurons, at least in this case, closely follow the control cycle. This is for PER2. And then the Schaff-Yang syndrome neurons that are here in green actually early on show a shift in their cycle relative to control and Prader-Willi. So we're in the process of adding more Schaff-Yang lines to this analysis and more deletion lines, and hopefully we will be able to get a handle on these circadian defects in cultured DPSC neurons. And the last thing I wanted to talk about is some single cell data that we recently developed in the lab. This is using a technique that was originally called split-seq that is developed now by a company called Parse. And essentially, it allows us to freeze everything in place. So what we do is we fix the cells and then we're able to label them and cycle through the labeling process enough times that we can then bioinformatically put the cells back together again to find the single cells. I know that's not a very good explanation, so I encourage you to go find out about Parse and split-seq. But essentially, we generated some very clean, very interesting single cell data. And what we did here is we looked at two controls versus, two controls versus two Prader-Willi deletion lines. And the first thing I want to show you is that when we look for gene expression for exist and SNRPN, we find the following. So we knew we had two females and two males in this study. And if you look at exist, which is expressed from the X chromosome and silent, obviously, in males, you find that the two female lines show expression of exist and the two male lines do not. Then you look at SNRPN. SNRPN is in that imprinted region. So again, we had two PWS versus two controls. You would expect SNRPN to be off in the PWS lines and on in the control lines. And in fact, it is. So this was encouraging. We then dug deeper into this data to find out if there were differentially expressed genes at the single cell level that maybe could be an indicator of the underlying developmental delay. And indeed, we found a couple genes that were very interesting. So we found that FOXP1 and FOXP2, both transcription factors that regulate neurogenesis, were actually dysregulated. So FOXP1 is up in the Prader-Willi samples and down in the controls. It actually should be down at this point as the neurons develop, but as you can see, it's somewhat delayed. FOXP2 should be up, right? But it is not really expressed in the PWS neurons and it is up in the controls. Again, we can look at some neuronal markers that are involved in neurotransmission that are at the synapse. Some more mature neurons would be expressing these markers. When we look at Neurexin-1, we see there's really nothing going on in PWS, and Neurexin-2 as well. We don't really see gene expression here. But when we look in the controls, we see that both of these markers are up. So all of these cells were aged the same way. They were differentiated and they were matured for 4 or 5 weeks. And then we did these gene expression studies. So in conclusion, I hope that they've gotten the point across that we have a new source for patient-derived neurons that we've been using in our lab and maybe other people would be interested in using. We have greater than 200 of these DPSC lines now in our repository, representing 14 different syndromes. So collection of DPSC and ASD status can both be done remotely using the SCQ. And I showed you the kits that we send out to families for this collection. DPSCs express neuronal markers like MAP2 and glial markers like GFAP. We've verified expression that is consistent with the disease state for Prader-Willi syndrome, for Angelman syndrome, and for Dup15q syndrome. I encourage you to look carefully at these papers so you'll see more details as to what we're seeing in terms of gene expression in these neurons. And then we use the DPSC neurons to investigate both DPSC molecular changes and PWS cellular changes that are associated with autism spectrum disorder. We showed that the autism-associated transcription factor BORA is upregulated in DUP15Q neurons. And we showed that there are ASD-associated mitochondrial defects in PWS-UPD neurons, at least in the individuals who score on the SEQ as being possible ASD. So we're currently studying circadian defects in neurons from SYS individuals, but we're also doing some additional single cell experiments, and recently we're funded to do more of that to try and flesh out these developmental defects that we see in PWS neurons. So I just want to put this up so you guys know you can really share this with families. Anyone who has a child that has a neurogenetic disorder, a syndrome with autism involved, they can contact us. And especially if we're collecting those particular syndromes, we'll send them a kit if they send us a genetics report. And hopefully, we can collect some teeth. It depends on how old these kids are. And then we will add them to our DPSC repository. So the majority of this work was done by a very talented graduate student in my lab, Caitlin Victor, who has been with me for a while and is hopefully going to graduate sometime next year. She just recently published her first primary research paper, so she's very excited about that, but we're going to get these other papers out, hopefully, on these other things she's been working very hard on. And I need to thank not only the former lab members like Nora Araka for these studies and Sarita Gurha, but also my collaborators, Janine LaSalle, who I mentioned, and Ryan Potts and Marty Donaldson, and some of these other people who've been involved in us doing these dental pulp stem cell studies. So with that, I will show you my funding sources and thank the Foundation for Prader-Willi Research, who has been really instrumental in driving these Prader-Willi projects forward. And I will hopefully take some interesting questions.